As we talk about the light reactions of photosynthesis, these will be taking place inside the thylakoids, but specifically there will be assortments of pigments within the thylakoid membranes. These pigment assortments are called light harvesting complexes because that is their job. It will be a bunch of pigments whose sole purpose is to collect as much light, as many colors of light as they possibly can. If we look at this picture here, in this picture we're seeing a light harvesting complex, and all of these green molecules are representing pigments. Now these are various pigments. Some of them may be chlorophyll A's, some may be chlorophyll B's. There certainly will be some carotenoids there as well. And they're going to be just kind of scattered out around this central pair of chlorophyll A molecules. So while the ones that are surrounding, so shown in the dark purple here, can be all different types of pigments, the two in the center will always be chlorophyll A molecules. And those two chlorophylls are going to have a special role in photosynthesis. Now what will happen is that light will come in and hopefully it will strike one of these surrounding pigments. A lot of times those surrounding pigments are called antenna pigments because they do act like little antennas. They're going to collect the light energy, and when they do collect it, they will transfer that energy. So the black arrows that we're seeing in this diagram are representing the transfer of that energy from one molecule to the next and so on. And eventually that will get transferred to the chlorophyll A pair. Once the energy is transferred to the chlorophyll A pair, an electron will get excited, so it will be boosted to a higher energy state, at which point it will be very unstable. So we must have a molecule that's going to be ready to collect that electron while it is still in a high energy state so that it won't fall back down to that ground state. So that molecule that will do the collecting is what we call the primary electron acceptor, and that we see right here. The primary electron acceptor will grab that electron and then it will pass it on, and we'll look at those reactions in just a moment. The whole purpose of the photosystem is to get the light energy and transfer it to the chlorophyll A pair. So we want to continuously collect energy and basically transfer it around until it gets to the chlorophyll A pair. Other ones will collect energy and transfer it to the chlorophyll A pair. So we're going to continuously be transferring the energy to the chlorophyll A pair. Now within the thylakoid membranes, we do have two different photosystems. The difference between them is the slight difference in the pigment assortments that they contain, and then they will have a different overall role in photosynthesis. Photosystem II does function first, and Photosystem I functions second. Now the names are based on the order they were discovered, so it is opposite of the order they actually function in. And because they do have different pigment assortments, they do have ideal or best wavelengths that are slightly different from each other. So sometimes they are named according to that part of it. Sometimes Photosystem II is called Photosystem 680 or P680, and Photosystem I sometimes called P700. If we look at exactly what's going to happen during the light reactions, they will involve the photosystems, and those photosystems will be collecting light energy, transferring it to that chlorophyll A pair that we looked at, and then there's two possibilities as far as what can happen to that particular electron once it is boosted up and collected. We have a linear route or non-cyclic electron flow, and then there's also a cyclic electron flow. Now we'll talk about both of these, but we will be talking about the non-cyclic electron flow because that is going to be the primary pathway that the electrons will take during the light reactions. So if we draw this out, here on this slide I'm going to have my thylakoid membrane across the middle, and we are going to have a lot of molecules embedded in this thylakoid membrane. Now down here on the bottom will represent my lumen, so that would be the inside of the thylakoid, and up here on the top this is going to represent my stroma. So that will be all the fluid surrounding the thylakoid. Within this thylakoid membrane, we will have our photosystems embedded. So this right here is going to represent my photosystem II. With non-cyclic electron flow, we do begin with photosystem II. Photosystem II does have all of those pigments scattered out as we just looked at. And then right in the middle, we're going to have that pair of chlorophyll A molecules. So to start us out, light is going to come in and hopefully strike one of these pigment molecules. When it does, they will collect that energy if it is of the right wavelength, and they will transfer the energy around until they get that energy to chlorophyll A. When it gets to the chlorophyll A pair, they will absorb the energy and the electron will get excited. So that electron will get boosted up to a higher energy state, and when it does, we have that other molecule that was ready and waiting, the primary electron acceptor, which is going to grab hold of that electron before it falls back down to the ground state. Now we'll come back to that electron in a moment. Notice that light's going to keep coming in and striking these pigments. So we're going to keep transferring energy to the chlorophyll A pair as much as we possibly can, which means that ideally we want to continue to boost electrons up and have those collected by the primary electron acceptor. If this process were to continue, and it's going to be taking place all day long, then we would need a mechanism to replace the electrons in chlorophyll A, because we would very shortly run out of electrons. This is where the water is going to come into play. If we look at our overall equation, keep in mind that we've got the water here, and we said that the water is going to get oxidized. So the water is going to lose electrons in this process. The reason the water is losing the electrons or getting oxidized is because it will be used to replace these electrons that keep leaving the chlorophyll A pair. So there will be an enzyme right here next to photosystem II whose job is to basically take a water molecule, oxidize it, and hand the electrons to chlorophyll A. So we're going to, right at the beginning of the light reactions, in non-cyclic electron flow, oxidize our water molecule and get those electrons. When we initially collect the electrons from the water, they will be low energy electrons. But notice with the light energy coming in, we are boosting them up and they are very quickly becoming high energy electrons. So valuable electrons at this point in time. Now as I've drawn it on here, we're going to use this as well to represent our free energy or energy scale. So notice we have boosted up the electron, so it is higher on the slide or on the diagram. High energy state. We're then going to take that electron and we're going to pass it down an electron transport chain. So we will have a collection of molecules embedded in this thylakoid membrane that make up an electron transport chain. So through a series of redox reactions, we will pass that electron down the electron transport chain, and notice at each redox step that electron will be losing a small amount of energy. As that energy is released, it will be used to do a very similar thing to what we did in the mitochondria with the electron transport chain. This electron transport chain is going to generate a hydrogen ion gradient. Now we're going to be using hydrogen ions, and the lumen generally has a high hydrogen concentration. The stroma tends to have a low hydrogen concentration. So the electron transport chain is going to generate the hydrogen ion gradient, so it will be moving hydrogens actively. If it is moving hydrogens actively, it needs to move them from low to high. In other words, the electron transport chain will be picking up hydrogens from the stroma and transporting it down to the lumen. Okay, so we're moving them then from low up here down to high. We're able to provide the energy for that active transport with the energy that is being released from this electron. So now we've got this electron sitting at the end of the electron transport chain. We've basically drained it of energy as we passed it down that electron transport chain. We'll come back to that particular electron in just a moment. we want to point out that the thylakoid membrane does also contain an ATP synthase enzyme. So if I draw my ATP synthase enzyme here, this one has the same structure as the ATP synthase that's located in the inner membrane of the mitochondria. So in other words, this one does have a tunnel that allows hydrogens to flow through across the membrane, and it does have active sites that are capable of taking ADP and phosphate and combining them to make ATP. So our hydrogens will flow through the ATP synthase, and that would be a facilitated diffusion. So at this point, they will be moving from high to low, which is the direction that they want to move. And when they do that, that will cause the wheel of this enzyme to turn, and that means that we'll be able to take our ADP plus phosphate and we'll be able to turn that into ATP. What we see happening over here is actually chemiosmosis. So we do have chemiosmosis take place in the light reactions of photosynthesis in a very similar manner to what we had in the mitochondria. One of the products of the light reactions is to make some ATP. So here we have taken light energy and used that energy to make our ATP because we boosted the electron up directly with the energy, but then we extracted the energy to power that ATP synthase. Now also in the thylakoid membrane, we are going to have our other photosystem. So this will be Photosystem I, so PS1. And Photosystem I has a similar arrangement to Photosystem II, just slightly different pigments. It does also have a pair of chlorophyll A molecules right in the middle. We will have more light come in to the leaf, so continuously coming into the leaf, and that energy will be transferred to chlorophyll A. When that energy is transferred to chlorophyll A, just like before, we get the electron that is boosted up, and that electron gets grabbed or accepted by a primary electron acceptor. Now this time, because again we're going to continuously transfer energy to our chlorophyll A pair and boost electrons up, we need to replace those missing electrons. That's exactly where this electron that we've just drained of energy is going to go. So since we're going to take the electron from the bottom of the electron transport chain, we will not have a need to oxidize another water molecule here. Now this electron that is now a high energy electron is going to be passed down a very short electron transport chain so that eventually it's going to be grabbed by NADP plus to then become NADPH. So NADPH will be holding on to an energetic electron, high energy electron. And if we look at the products of these light reactions, this non-cyclic electron flow, we did produce some oxygen gas over here. Oxygen gas is a waste product, and we can see that here in our overall equation for photosynthesis. We also produced the necessary reactants, some of the necessary reactants for the Calvin cycle, and that's the NADPH and the ATP. The NADPH is going to provide the energized electron that is necessary to reduce the carbon dioxide, and the ATP will provide the chemical energy that will be necessary to do that. I want to emphasize that both of those products, the NADPH and the ATP, they are produced and now located in the stroma of the chloroplast, which is an ideal location because that is exactly where the Calvin cycle is going to take place. If we talk about, or first we just look at this diagram right here. This is a picture from your book that simplifies this process we just looked at. Essentially, we're going to have light come in. We're going to excite that electron, which originally does come from a water molecule. And then we're going to drain that electron of its energy. As we do that, we will be making ATP by chemiosmosis, and then we will be reboosting that electron up with additional light energy, and it will go to NADPH. Notice when it is passed to NADPH that that electron is still at a very high energy state. So it's an energetic electron, which will be used for the Calvin cycle. If we look at the alternative pathway for electron flow, this is going to be a cyclic pathway. Cyclic because they are taking a circular route. This particular pathway is only going to use Photosystem I and is only going to produce ATP. So we do have different products being produced in this second method. If we look at this diagram here, we can see that it is still going to be the thylakoid membrane involved. We'll have Photosystem I embedded in that membrane. The light will come in, it will strike a pigment. When it strikes this pigment, the energy will be transferred until it gets to that pair of chlorophyll A's. When it gets to the chlorophyll A's, the electron is boosted up, it is collected by the primary electron acceptor, and then it will be passed down an electron transport chain. That electron transport chain will move hydrogens across the membrane to generate the hydrogen ion gradient, and it will power the ATP synthase so that we will get ATP made by chemiosmosis. Now this is a cyclic electron flow because once we have drained all the energy out of that electron by the electron transport chain, we're going to hand that electron right back to the chlorophyll A. So the electron is going to come from the chlorophyll A and it's going to go right back to the chlorophyll A. Notice that we do not oxidize any water in cyclic electron flow, which means that we will not produce any oxygen gas. And then additionally, we don't have those NADPHs being made. So when we look at those two, you may wonder why there's going to be two different types of electron flow. Well, first of all, it should be obvious that they are producing different things at the end. Non-cyclic electron flow produces roughly equal quantities of NADPH and ATP, but the Calvin cycle does actually require more ATP. The whole purpose of the light reactions is to supply the needs of the Calvin cycle. So it's really all about balancing our supply with the demand. Because we need more ATP, we have to have a method for making up that extra ATP. And that's where the cyclic electron flow is going to come into play. So most of the time, the thylakoids will run the non-cyclic electron flow, and they will make lots of NADPH and lots of ATP. When they need some extra ATP made, they will switch into the cyclic electron flow mode, make that extra ATP, and then switch right back to the non-cyclic electron flow. So we will constantly be switching back and forth just to provide whatever the Calvin cycle is going to need. Now another thing we want to really emphasize here, which we've mentioned, is that we do have chemiosmosis taking place during the light reactions. So we have chemiosmosis happening in the mitochondria with the process of cellular respiration during the oxidative phosphorylation, and we also have chemiosmosis during the light reactions of the chloroplast. And that was taking place with both the cyclic electron flow and the non-cyclic electron flow. So we want to be very familiar with how these two chemiosmosis processes compare with each other. As far as the similarities are concerned, both of them contain an electron transport chain embedded in the membranes. So in the mitochondria, it happened to be the inner membrane of the mitochondria, and in the chloroplast, it's the thylakoid membrane. Both processes are going to involve an ATP synthase complex that has essentially the same structure, and they have similar electron carriers. The electron carriers would be the specific molecules that are doing the passing of the electrons throughout all of this. And those electron carriers are going to be capable of generating that hydrogen ion gradient. Now, as far as the differences are concerned, the mitochondria get the energy for powering their chemiosmosis from organic molecules. That specifically, in the example we looked at, came from glucose. But it could come from other food sources. So it could come from proteins, it could come from fat molecules, steroids for that matter, but it's coming from an organic molecule, chemical energy. The chloroplast, on the other hand, is going to use light energy, so it's collecting the light and using it to make ATP. In both cases, you do have this temporary holder of the electrons, and it's NADH in the mitochondria, it's NADPH in the chloroplast. Only difference between the two is that phosphate group that we have in the NADPH. We also want to be aware of the spatial organization that we're dealing with. When we're looking at the chloroplast, in the thylakoid space, or the lumen, is where you're going to have the very high hydrogen concentration. And then you're going to have a low hydrogen concentration in the stroma. So very similar to what you see in this diagram. So we want to be aware that the electron transport chain will be taking the hydrogens and it will be moving them into the thylakoid space. As they move out through the ATP synthase, they're going to be going right back into the stroma. Notice how we are cycling those electrons around. If we want to continually make ATP, we've got to be able to continually bring those hydrogens back into the thylakoid space, which is what that electron transport chain was doing. Now, if we look at the mitochondria spatial organization, keep in mind that in the mitochondria, we had two spaces. And in the mitochondria, it was the intermembrane space that had your very high hydrogen concentration. In this matrix, we had low. So in this case, the electron transport chain was actually moving the hydrogens into the intermembrane space, and then as they pass through the ATP synthase, they go right back into the matrix. Again, make sure we understand the spatial organization, which direction the hydrogens are being pumped by the electron transport chain, and which direction they are flowing when they pass back through the ATP synthase. So here's a good representation of this. This picture shows where the high hydrogen concentrations are at. It's in the shaded, darkly shaded areas. So it's around the outside in the case of the mitochondria, and in the inside in the case of the chloroplast. So they're somewhat opposite of each other. We should also notice that in the mitochondria, we're getting our ATP produced in the matrix, and in the chloroplast, it would be produced in the stroma. This diagram right here gives us a good summary of the non-cyclic electron flow to really sum up our light reactions. In non-cyclic electron flow, we've got light coming in to photosystem II. Hopefully that light is going to strike a pigment. They will transfer that energy until it gets to chlorophyll A. When the light gets to chlorophyll A, we are essentially taking an electron from water and we're going to boost that electron up to a high energy state. That electron will be collected by a primary electron acceptor and then passed down an electron transport chain. Notice as we pass the hydrogen or the electrons down the electron transport chain, we will be picking up hydrogens from the stroma, transporting them into the thylakoid space. As long as we can keep that electron transport chain going, we're going to generate that hydrogen ion gradient. When we get to photosystem I, that electron will be reboosted up with additional light energy, and then it will be passed to NADPH in the stroma. We do also have the chemiosmosis taking place at the same time as all of this, because those hydrogens are going to flow back out through the little channel in the ATP synthase. As long as the electron transport chain continues to run, the ATP synthase will continue to run. There are some herbicides, things that specifically kill plants, that do affect that particular electron transport chain. If you shut down that electron transport chain, you do away with the plant's ability to use light energy. So for them, they would be able to absorb it with the pigments, but the electron transport chain doesn't generate the hydrogen ion gradient. Those herbicides specifically inhibit plants and not animals because animals don't have that specific electron transport chain. The one in the mitochondria, although very similar to this one, is different enough that it's not affected by those same molecules. So that sums up the light reactions. In the light reactions, we want to make sure we understand where everything is taking place, what the difference is between Photosystem I and Photosystem II, and then the difference between the two types of electron flow. The second stage of photosynthesis is the Calvin cycle, and the Calvin cycle is going to take place in the stroma. So here we're looking at the area surrounding the thylakoids. This is an ideal location because it's going to take the ATP and the NADPH, which are already in the stroma, and it will be using those to actually reduce the carbon dioxide into sugar. When we reduce the carbon dioxide into sugar, we will produce a small carbohydrate, which will be a 3-carbon carbohydrate, which is glyceraldehyde 3-phosphate, or what we typically call G3P. That molecule should look somewhat familiar to you because it is a molecule that we saw during glycolysis. Because it is a 3-carbon sugar, it will require the use of 3 molecules of carbon dioxide to make that particular sugar. In this process, the Calvin cycle, we are essentially taking our inorganic carbon and we are converting it into organic. Very important process here. This process is what we call fixing the carbon. So the Calvin cycle's job is to fix the carbon. Basically take inorganic carbon and put that into a useful form, and a form that heterotrophic organisms are going to then be able to utilize. We will have an enzyme at the very beginning of this process that is critical for the fixing of the carbon, or that does the initial fixing of the carbon. And this enzyme's name is going to be Rubisco. Rubisco is the most abundant enzyme, or thought to be the most abundant enzyme that we have on the Earth's surface. And this is the enzyme that's responsible for converting all of that carbon dioxide back into a form that we can then consume ourselves. So it is a very, very important enzyme. If we look at the Calvin cycle is a cycle. So this means that it needs to continuously run. And just like we had with the citric acid cycle, we're going to have one molecule that is ready and waiting to collect or bind to a carbon dioxide once it enters the cycle. So this molecule is ribulose bisphosphate. Ribulose bisphosphate is a 5-carbon molecule. It's going to combine with a 1-carbon carbon dioxide and will end up with a 6-carbon molecule. Notice right here at the beginning of this, we're going to have our Rubisco enzyme. Okay, so Rubisco is going to catalyze the very first step of the Calvin cycle. The name Rubisco, first of all, starts with the substrate that it uses, ribulose bisphosphate, and then it also has that co part on the name. That co part stands for the 2 functions of Rubisco, which happen to be carboxylase and then oxygenase. So Rubisco actually has 2 different functions that it can perform depending on the substrate that it binds. In the Calvin cycle, it will be binding the carbon dioxide. So when it binds carbon dioxide, we're going to get the carboxylase function. Okay, so that's what we're seeing in this diagram. We're seeing the carboxylase function. We will come back to the oxygenase function in just a little bit. So we're going to take 5 carbons, add to that 1 carbon, we get something that has 6 carbons. Now this first 6-carbon molecule is highly unstable and it's not going to last very long. So very quickly it's going to be broken down into 3-carbon molecules. So broken in half essentially. This 3-carbon molecule is the first stable organic molecule that we get from this process. So this that we're looking at is what we typically call C3 metabolism. This is what most plants would fall into or fall under. So most plants are going to have C3 metabolism when it comes to photosynthesis. We want to notice as well that the Rubisco enzyme is actually fixing the carbon, as we would call it, because it's taking inorganic carbon and it is converting it into an organic form. Now, if we continue to look at the Calvin cycle, we are going to take some ATPs and use those. Those would be ones that came from the light reactions. We will be reducing our carbon with electrons from NADPH. And then we do end up with our glyceraldehyde 3-phosphate. That glyceraldehyde 3-phosphate is a product. It can leave the cycle at that point. But then we do have the second half of the cycle, which is for the purpose of regenerating that ribulose bisphosphate. That will require some additional ATP from the light reactions. So we started with ribulose bisphosphate and we need to end with ribulose bisphosphate. Now, if we talk numbers here, if we want to make one net G3P, like we have down here, notice that this G3P has 3 carbons in it. So if we're going to make one of those, we need to use 3 carbon dioxides. So if we look numbers, we're going to take 3 ribulose bisphosphates, which would be 5 carbon molecules. So 3 times 5, we've got 15 carbons right here. We're going to add to that 3 carbons with our 3 carbon dioxides that we add to each of those. So we're taking 15 carbons plus 3 carbons, and we're going to end up with 3 6-carbon molecules, which is 18 carbons. We'll split all of those into 3 carbon molecules. So we've got 6 3-carbon molecules, which is still 18 carbons. And this will still be 18 carbons and down here as well. One of those G3Ps will leave the cycle as a product. So notice the G3P has 3 carbons, so we're going to take our 18 minus 3, and we end up with 15 carbons here. That would be in the form of 5 3-carbon molecules, and that 15 carbons will be used to get back to this 15 carbons here. So we are regenerating the RUBP, or the ribulose bisphosphate. If we are going to make a product that is 3 carbons in size, we need to use 3 carbons to do that, or 3 carbon dioxides. Now with the Calvin cycle, you will not have to count up ATPs and NADPHs in the same fashion as what we did with cellular respiration. However, we do want to understand that the Calvin cycle does definitely require more ATP than it does the NADPH. So for each glyceraldehyde 3-phosphate that is made net, the cell will spend 9 ATPs and 6 NADPHs. Keep in mind that the whole purpose for having that second type of electron flow, the cyclic electron flow, was to make extra ATP. So that right there just emphasizes why we did need to make extra ATP. Now, while we're ending with a glyceraldehyde 3-phosphate, you could certainly build larger carbohydrates from this if there was a need to. So we could take 2 G3Ps, so 2 3-carbon molecules, and we could build a glucose from that, so a larger monosaccharide. We could take it and make disaccharides such as sucrose, which is the primary transportable sugar inside of a plant. We could take the glucose and we could hook them together many times and make a starch molecule. So that's also a very common carbohydrate that we find inside plants. So although we're not looking at all of the steps and the enzymes involved in those conversions, we do want to be very much aware that just because we're stopping with the glyceraldehyde 3-phosphate, there are other carbohydrates that can be made. It just requires more steps.